Okay, so picking up where we left off, we are now going to go through the full order of events of an action potential from pre to postsynaptic side. All right, buckle up. So we talked about all the components, right? Presynaptic side, we have our nervous system component, which has our axon terminals, and at the end they each have this axon. It's called a synaptic bulb at the end. Here is our synaptic cleft, where within this bulb, we have the vesicles of acetylcholine. We have an inactive enzyme that free floats throughout this bulb at the end, as well as we have calcium on the outside of the cell, which is going to be a player in coming into these voltage-dependent gates right here. Remember I said we have down the axon terminal, we have myelin sheath, but then where the myelin sheath stops, we have voltage-dependent gates, and that's a key player with regards to calcium coming in. On this postsynaptic side, we have these troughs at the motor end plate. In these troughs, we have our receptors, which are alpha receptors, beta, gamma, and sigma, but we really care about the neuromuscular junction right now, and we really care about the alpha receptors because that is what acetylcholine is going to be looking for to attach to then now create an end plate potential and an action potential in this cell in order for us to contract. And we're going to get to muscular contraction after we talk about these steps. Okay, so moving forward, before we talk about these steps, I'm going to do my best to actually draw what I'm talking about as we go over the steps. So I'm hoping my stylus works okay for this drawing as I go through the steps with you. So hang tight. Okay, so here we have our axon terminal. That's our myelin sheath right here. And these are voltage-dependent gates right here. And this is our synaptic bulb at the end. And then within this bulb, we have those vesicles which contain a quantum each of acetylcholine. Right, so there's a bunch in there. We have what's called an inactive enzyme that is in here. Apologize for my handwriting while I'm trying to do this on the computer screen. Right, and then down here we have, these are what's called dense bars, which are at the end of the synaptic bulb. And then here we have our postsynaptic side, right? So we have our alpha receptors, which is what we care about, sitting on top. And then we also have our beta, sigma, gamma, beta, sigma, gamma, all that good stuff down here as well. But we don't really care about those for right now. But those are down at the bottom. And we also have more alpha receptors up and down these troughs. And again, we doubled the amount of alpha receptors, which is good because that's what acetylcholine is going to look for to bind to. Within here, that's our amorphous substance that is rich in carbohydrates. And then we have calcium on the outside right here. Calcium on the outside right here. Trying to see if I forgot anything, right? So those are our voltage dependent gates right here. And here we have our myelin sheath. So this is our presynaptic side, and this is our postsynaptic side. In case any of you were curious, I minored in neuromuscular junction drawing in undergrad. I'm just kidding, this is terrible. Okay, so let's go over the order of events for this entire action potential now. So we've generated an action potential after cellular depolarization has occurred, right? So we altered that on one side, on one cell, we altered the relationship between sodium and potassium. Now the signal, which is a moving wave of electrical current, will travel down the axon. So here we go. Moving wave of electrical current is traveling down the axon towards those axon terminals, towards the synaptic bulb. The charge inside the cell will go from negative to positive. And now what's a key player is these voltage dependent gates, as the signal travels down, these voltage dependent gates right here on the unmyelinated portion of the axon terminal, these will open up. So here we go. Let's do this. These gates are going to open up as this signal travels down. We are going to open up those voltage dependent gates. Next, as those gates are opening, we're going to take calcium, and calcium is going to move down its concentration gradient from the outside of the cell into the inside of this axon terminal, and it's going to come through those voltage dependent gates and come into the bulb down here. Calcium is going to come in down there. Calcium will travel down into the synaptic bulb. This increase in calcium concentration turns now this inactive enzyme to an active enzyme. So key player, calcium comes down into the synaptic bulb and now activates this inactive enzyme right here. This is the switch that turns on synaptic transmission. Now, next step, we have this inactive enzyme. What is that inactive enzyme going to do? That inactive enzyme is going to now trigger these vesicles of acetylcholine. They are going to migrate down to these dense bars. Going to migrate down to those dense bars or active sites. They're called, these things down here are called dense bars or active sites. Those dense bars, so let's use a different color now, let's use green. These dense bars are going to open up via exocytosis, and each vesicle of acetylcholine is going to be released into this cleft now. Now it is free flowing in this carbohydrate rich substance. Acetylcholine will travel now, once those dense bars open, acetylcholine will travel and find those alpha receptors to bind to, right? On this postsynaptic side. If enough alpha receptors are activated, the membrane permeability will be altered on this postsynaptic side. This permeability will now be altered. Now you can officially say you have generated an end plate potential. This has resulted from the change in membrane permeability of sodium and potassium that now triggers a transient wave of depolarization, which is followed by action potentials. So ultimately, now what are we doing? After we have altered the membrane permeability on the postsynaptic side and we have transmitted that signal from one cell to the next, now that signal can travel down the muscle cell. And then our next series of events is going to be having the cellular muscular contraction take place. Okay, so now we're back onto the PowerPoint. We just went over the order of events with what I drew with the stylus on the computer, and that is how we develop an end plate potential. And I have uploaded for you the order of events, which hopefully will help. Okay, now we're on the postsynaptic side and we're going to be strictly talking about skeletal muscle right now. So we're shifting gears and now talking about skeletal muscle, the structure and function. Skeletal muscle accounts for about 40 to 50% of a body's total mass, or our weight. At rest, skeletal muscles can receive about 25% of the total oxygen consumed. On average, we will consume 1/4 liter of oxygen per minute, or about 250 mLs. So if skeletal muscle gets 25% of that, that comes to about 62.5 mLs at rest. During exercise, the O2 being consumed by skeletal muscles can increase 20-fold in absolute terms, depending on the intensity of the exercise. So if we're going to give more O2 to the working muscles, what must happen to other physiological activities? We must decrease that activity. of certain organs and certain parts of the body. So what we're going to do is we're going to shunt blood from areas of less activity to areas of higher activity. So if we want more blood to come to our limbs and our muscles, what's causing us to either run, jump, swim, or do whatever, our skeletal muscles need more blood and nutrients. So in order for us to do that, we need to shunt blood from areas of less activity to areas of higher activity. So where are some areas we would shunt blood from? We would shunt blood from most of our splanchnic region, which is in our core. So our digestive system, we'll borrow things from our stomach, our kidneys, our intestines, and we'll talk about what we do to keep those tissues viable next as well. We will talk more about shunting blood from areas of less activity to higher activity when we talk a little bit more about cardiovascular hemodynamics, and vasodilation and constriction later on in the semester. So next, let's still continue with the structure of a muscle. So we have the full skeletal muscle, right? So let's talk about one full muscle would be right here. We have different fascicles. We have individual muscle fibers within those fascicles, and then we have myofibrils within those muscle fibers as well. We also have myofilaments, which are in those myofibrils, and those are actin and myosin, right? All of these structures are embedded in a sarcoplasmic gel, which is our cytosol, right? Okay. So each individual muscle fiber is wrapped in a connective tissue called the endomysium. Individual muscle fiber. So if we come here and we go into an individual muscle fiber, it is wrapped in endomysium. A fasciculus, so over here, so this would be this is one muscle fiber, so this would be the endomysium right here, wrapped around one muscle fiber. Here is a fascicle of muscle fibers that is wrapped in perimysium. It's also another layer of connective tissue. And then the epimysium wraps around the entire muscle. So for example, it's consistent with a tendon for the most part. So let's say right here was, a deltoid. This whole deltoid muscle is wrapped in an epimysium. Each fascicle, which contains multiple muscle fibers, are wrapped in a perimysium, which this would be one fascicle. And then each individual muscle fiber is wrapped in something called an endomysium. Okay, so now that we know each component to a full muscle fascicle, muscle fiber, we care about the actual contractile component to muscle cells. These are our thick and thin filaments. So in order for us to generate force, we must slide the thick and thin filaments over each other. Our thick filament is our myosin, which is right here, and our thin filament is our actin, right, which are over here, these little pearl-like type of things. The more cross bridges we have, the more force the muscle fiber can generate. Makes sense, right? So the more of these connections we have, the more we can overlap and pull on each other, and we will generate more force. Let's talk about our thin filament for a second, which is our actin. These are composed of a double helix strand. You can see down here, double helix, and they twist within each other. Just if you had a string of pearls and then you twisted the ends, that's what you would get. Each string of pearls, so each string of actin is called an F-actin strand. Each individual pearl right here, each individual actin pearl is called a G-actin subunit. And each G-actin subunit has a myosin binding site. So all of these little actin molecules here, which is the G-actin subunit, each one of those, you can see G-actin there, has a site for which myosin can bind to that. So actin has two regular proteins associated with it, tropomyosin, which is this orange structure right here in our double strand, and troponin. And troponin consists of 3 molecular subunits, which is right there. Each subunit will bind to something other than the other two, actin, tropomyosin, and calcium. We will continue this in the next video.